Hi, my name is Lisa, and I am a biologist at the Public Health Agency of Canada. I'll be talking about my poster on the genomic analysis of antifungal resistance in Candida albicans in Canada from 2012 to 2020. Candida albicans is the most common fungal pathogen in invasive infections. It has been declared by the World Health Organization as a critical priority fungal pathogen. Few drug classes are available for treatment in Canada and include azoles, echinocandins, and polyenes. The problem is multi-drug resistant isolates with antifungal resistance to more than one class of drug have already been identified. This particular study, we aim to examine the strain distribution and mechanisms of resistance of C. albicans using whole genome sequencing of 137 human clinical isolates from Canada. The collection was enriched for resistant and invasive isolates, and potential mechanisms of resistance were examined by identifying known and novel mutations in 15 key genes previously implicated in antifungal resistance. So a bit of background. I have a diagram of a fungal cell with the 15 genes in red text that were previously implicated in antifungal resistance. Antifungal drugs such as azoles inhibit lanosterol 14-alpha demethylase in the ergosterol synthesis pathway encoded by ERG11. Additional genes ERG2, 3, and 5 include enzymes in the pathway and may lead to resistance via altered drug target. Polyene drugs extract ergosterol from the membrane, and echinocandins target beta-glucan synthase encoded by FKS1 and FKS2. Additional mechanisms of resistance include upregulation of efflux pumps through increased expression of ATP binding ABC transporters encoded by CDR1, CDR2, SNQ2, and ABC1, and major facilitator superfamily MFS transporters, MDR1 and TPO3. Transcription factors, MRR1, TAC1, and UPC2 may also activate expression of transporters, thereby decreasing intracellular concentrations of antifungal drugs, which have been denoted using the pink circles in my diagram, producing a resistant phenotype. We collected 137 C. albicans clinical isolates, 20 from Western Canada, including British Columbia and Alberta, 19 from Central West, which includes Saskatchewan and Manitoba, 70 from Central East, which encompasses Ontario and Quebec, and 28 from Eastern Canada, including Newfoundland and Labrador, Prince Edward Island, New Brunswick, and Nova Scotia. Once we received the isolates, we plated them on sabro-dextrose agar and completed DNA extraction using the Epicenter MasterPure Complete DNA and RNA Purification Kit, then the Nextera XT-DNA Library Prep Kit and NextSeq 1000-2000 P1 kits for whole genome sequencing on the NextSeq 1000-2000 Illumina platform. The same isolates were screened for resistance through antifungal susceptibility testing based on the CLSI guidelines with custom panels from Thermo Fisher. The panels were then incubated, and the minimum inhibitory concentration was determined for fluconazole, micafungin, and amphotericin B. Once the whole genome sequencing data was produced, phylogenomic analysis was completed based on homozygous single nucleotide variants, otherwise known as SNVs, in the core genome using the SNVL pipeline version 1.2.3 with C. albicans SC5314 as the reference. Resistance variant analysis based on SNVs was completed using a custom pipeline by Anna from Université Laval in Quebec. And our statistical software with open source packages was used for annotation and visualization of the phylogenomic tree. This is the phylogenomic tree of the 137 clinical C. albicans isolates from Canadian hospitals. You'll notice that we have the isolation site grouped into broad categories, including non-sterile or sterile. Non-sterile includes oral cavity and pharynx, other and vagina, and sterile includes blood and other invasive isolates. These are all denoted by triangles, and the regions are denoted by circles and include Central East, Central West, East, and West. If the isolates were azole resistant, they were also highlighted in blue, and if they were echinocandin resistant, they were highlighted in green. You'll notice right off the bat that we have here cluster 1 of interest. These isolates all significantly correlated with azole resistance and the Central East region of Canada, which includes Ontario or Quebec, and were primarily vaginal isolates. Phylogenomic analysis revealed a high level of diversity, with isolates differing from the reference by 420 to 5,246 SNPs. Of the 137 resistant isolate-enriched C. albicans collection, 54 isolates were fluconazole resistant, 8 isolates were micafungin resistant, and all isolates were wild-type for amphotericin B. We were able to detect 46 potentially novel variants only present in resistant isolates for 14 of the 15 genes. The majority of azole-resistant isolates contained a known substitution in ERG11, and 14 fluconazole-resistant isolates and 1 micafungin-resistant isolate lacked any known or potentially novel substitutions in the genes examined. Cluster 1 of interest, which is composed of 13 primarily vaginal isolates, correlated significantly with azole resistance and the Central East region of Canada, which is Ontario or Quebec. We identified 151 novel natural variants which are present in both susceptible and resistant isolates. In Table 1, we have all of the variants that we identified in fluconazole or micafungin-resistant isolates, either highlighted in blue or green, and we've grouped them into 3 categories. Confirmed variants, which are those that were previously experimentally verified to confer resistance in C. albicans. Suspected variants, which were not experimentally verified, but were found in at least one other resistant isolate from a previous study. And potentially novel variants, which are those that were identified for the first time in resistant C. albicans isolates in this study. I have stars next to some of the numbers indicating that each variant only occurred once to save space, and also some brackets around variants indicating that these all occurred in a single isolate. All in all, this analysis revealed an overall high level of genetic diversity in C. albicans. A variety of genetic mutations potentially conferring antifungal resistance were detected, which may contribute to future molecular identification of resistance. I'd like to thank the National Microbiology Laboratory Branch's Genomic Core Facility and Bioinformatics Core Facility for providing whole genome sequencing and bioinformatics support. These are all my references. Thank you so much for listening to my poster.